Good afternoon, students. In previous class, we discussed outbreeding devices. So, outbreeding devices which encourage cross-pollination, which prevent self-pollination. So, those devices which are called as outbreeding devices, which are unisexuality or monocliny, heterostyle, herpogamy, self-incompatibility, like those 5 outbreeding devices we studied in last class. So also a little bit we discussed that is a pollen-pistil interaction. So in pollen-pistil interaction, there is a number of events are there. Those events are pollination, pollen germination, pollen tube growth through the style towards the ovary, entry of pollen tube into embryo sac. So these all events which together which are called as that is pollen pistil interaction. So we discussed one by one events of these pollen pistil interaction. So first one, that is pollination. What is pollination? Already we discussed in previous classes. So transfer of pollen grain from anther to stigma of either same flower, stigma of another flower present in the same plant, or stigma of another flower present in another plant. So those three we call it as that is pollination. So there is successful pollination either by a different mediators, a different medium, either water or wind or any other animals. So like the pollination is completes, it is nothing but transfer of pollen grain from anther to stigma of either same flower or another flower. So that is what we are called as pollination. Second one, pollen germination. So whether the stigma is that is landing surface of pollen grain. The pollen grain fall on stigmatic surface of a pistil. So during which these pollen is accepted or rejected both either based on that is the chemical analysis of these pollen grain and chemical analysis of the stigmatic surface, because chemicals like the enzymes, proteins, lipids, carbohydrates, those which are present on the stigmatic surface, and also certain enzymes which are filled in the germ pores of these pollen grain. The pollen exile is made up with sporopollenin and also presence of proteins and carbohydrates on these. Based on these chemical analysis of pollen grain and stigmatic surface, the stigmatic surface is identified. These pollen is compatible or non-compatible. Compatible or non-compatible. If the pollen is compatible, this stigmatic surface is allowed to germination of these pollen grain. So the germination is nothing but there is protruding or emerging a pollen tube from pollen grain. So at the time of pollen germination, either one pollen tube or many pollen tubes are also emerged from a pollen grain. In most cases, the pollen grain which emerge a single pollen tube from their germ pore. So in extraordinary cases, the pollen which emerge 5, 3, 10, 14, like different a number of pollen tubes which are emerged from a single pollen also. While pollen germinate, what it emerged the pollen tube? The pollen tube emerged by the intine of pollen grain. At the time of germination, there is a plug which emerges out through a germ pore, during which the swelling of that pollen grain, during which the intine layer which emerges from the germ pore. At the time of emerging, that pollen tube which consists whether the pollen grain, it is the exile, it is the intine only which swollen and which emerged out through the germ pore, during which the cytoplasm of pollen grain is entered into pollen tube. Two male gametes is also entered into pollen tube, and a vegetative nucleus is also present in the pollen tube. So means in this pollen tube there is presence of cytoplasm. These two are male gametes. Two male gametes are emerging, and there is vegetative nucleus. Totally moving into the pollen tube. It is nothing but the contents of pollen grain entered into pollen tube. So as these pollen tube emerge from those pollen grain, it is what we call it as that is totally pollen germination. If pollen grain is non-compatible, so compatibility is nothing but the pollen grain of the same species, if maybe of other plant or same plant. Non-compatible is nothing but the pollen grain of other species. Other species maybe fall on these. The stigmatic surface is rejected. So at the time of pollination, certain stigmatic exudes, because the stigmatic surface is wet condition. Due to wet condition, it is nothing but certain exudates is secreted by the cells of the stigmatic surface. Those exudes which secrete on the surface, it helps to wetting nature of the stigmatic surface. So the wetting nature of the stigmatic surface is one of the advantages for the pollination, one of the advantages for the pollen grain, because the pollen grain is firmly attached to the substratum of the stigmatic surface. As the pollen, that is the stigmatic exudates, is also helps to, that is attachment of pollen grain on stigmatic surface. So this wetting nature is also required for the germination of pollen grain. So because the pollen grain, it is little, that is pollen surface is little bit dried nature, non-sticky. So during which the wet condition helps to sticky nature. That exudates, which is also consists certain nutrients, that exudates will also helps to pollen germination, because pollen germination, it required certain moisture content, certain that is water content, it is absorbed by these, what the wet nature of these stigmatic surface. So those exudes which is also helps to, that is pollen germination. After next, the third event, that is pollen tube growth through the style. So here already we know that is The pistil, it consists of three important parts, that is stigma, a long style, and basal bulged portion, it is called as ovary. So here a pollen tube, a pollen germinate, the pollen grain is germinate. At the time of germination, the pollen tube emerged from the pollen grain. These pollen tube which grow towards the ovary, as in the ovary there is presence of ovule. So here, if the style is, there are three types of styles, either solid, either open or hollow, either semi-solid. If solid, it is nothing but it is totally filled with a tissue. The tissue which is having a long cells in the style. Either open or hollow. It is nothing but there is presence of a inner layer only. So there is a hollow space. Through the space, the pollen tube is easily moved. Otherwise, semi-solid nature. Semi-solid nature is nothing but here in the center there is presence of little bit more tissue, but even though there is presence of a small hollow space. Like the three types of styles are there, through which, if any type of style, the pollen tube which grow towards the ovary. The pollen tube emerged from the pollen grain which entered into stigmatic surface. For example, here the stigmatic surface, especially the pollen tube entered into stigmatic surface through the intercellular spaces of the a layer of cells present in the stigmatic surface. So ಇಲ್ಲಿರ್ತಕ್ಕಂಥ cells ಅಲ್ಲೇ intercellular spaces through which the pollen tube which is entered into the stigmatic surface. Stigmatic surface ಮುಖಾಂತರ pollen tube ಒಳಗಡೆ ಬಂದ ತಕ್ಷಣ, so once again these pollen tube which grow towards the ovary or towards the ovule. So as the pollen tube growth through the style towards the ovary. If number of pollen tubes, that is the pollen grain, a number of pollen grains are germinated on the stigmatic surface. If all the pollen tubes which grow towards the ovule, which is a pollen tube which first entered towards the ovule, it must enter. So that is the entry of pollen tube into embryo sac. Entry of pollen tube. There is a path of pollen tube. As according to the way of pollen tube entered into embryo sac, there are three paths, or the three types of that is porogamy, mesogamy, and chalazogamy. Porogamy, mesogamy, chalazogamy. As it is depend upon the entry of pollen tube into embryo sac. Here it is a pollen tube the pollen tube which entered into ovule through micropylar pore. So it is the micropyle. Through the micropyle only, the pollen tube entered into embryo sac or into a ovule through the micropyle. This phenomenon is called as porogamy. Porogamy is nothing but entry of pollen tube entered into embryo sac through the pore. The pore present in ovule, that is called as micropyle. That pore is nothing but it is not covered with two integuments, through that pore the pollen tube entered into embryo sac. So this type of entry of pollen tube into embryo sac, it is called as porogamy. So second one, mesogamy. So mesogamy is nothing but the pollen tube which entered into embryo sac through the either integuments or otherwise either through funicle. But once again they entered along through the micropylar region. So, but these pollen tube entered through the integuments. Already we know that the ovule consists either single or two integuments, bitechnic ovule or unitechnic ovule. Through the integuments, the pollen tube is entered into the ovule. So this type of phenomenon or process, what we are called as mesogamy. So it is the mesogamy. So in mesogamy, pollen tube enter into embryo sac through either integuments or either funicle. The funicle, it is nothing but stalk of the ovule. Stalk of the ovule, it is called as funiculus, that is funicle. So ಇಲ್ಲಿ pollen tube integuments ಮುಖಾಂತರನು ಎಂಟರ್ ಆಗಬಹುದು ಅಥವಾ funicle ಮುಖಾಂತರನು ಎಂಟರ್ ಆಗಬಹುದು. So it is this phenomenon what we are called as mesogamy. The third one is chalazogamy. So here the pollen tube entered into the pollen tube. That pollen tube entered into the chalazal part of ovule. What is chalaza? The broad base of ovule. The broad base of body of ovule, it is called as chalaza. So as we know that there are two endings, chalazal ending and micropylar ending. Here the pollen tube entered into embryo sac through the chalazal part of ovule. That's why such phenomenon what we are called as, that is chalazogamy. Gamy is nothing but fusion of male gametes with the contents of two different contents of embryo sac. So as this phenomenon what we are called as chalazogamy, the entry of pollen tube into embryo sac by three ways, that is orogamy, mesogamy and chalazogamy. Orogamy means pollen tube entered into embryo sac through the micropyle. It is through the chalazon. So mesogamy, the pollen tube entered into embryo sac through either integument or funicle. Chalazogamy, that is, see the pollen tube entered into embryo sac through chalaza of the ovule. So these all events which considered as, that is, pollen pistil interaction. Pollen pistil interaction. So next, artificial hybridization. Artificial hybridization. What is hybridization? Here, hybridization is nothing but the process of crossing of two plants, crossing of two different plants, which differ genetically one or more characters. One or more characters. Such process what we are called as hybridization. Hybridization. In artificial hybridization, we are using to improvement of crop, to improvement of crops. So artificial hybridization is one of the unique process. It will help to improvement of any crop. Here we have taken two desirable characters. So these two desirable characters present in two different plants which combine in the same plant. So it is a little bit helpful to improve the crops, to improve the nutrients in the food, or that is it is nothing but food crops. So as these hybridization, it is successful by two important processes, that is emasculation and bagging. These two events which are must and should require to successful artificial hybridization. In second P only we discussed, that is in plant breeding, the plant breeding techniques, in which those plant breeding techniques, first we selected the two different plants, those as a parental plants, that is selection of parental plants. So one used as the male parent one used as female parent. So at the time of selection of parents, those two parental plants which is having desirable characters. While the desirable characters, for example, there is presence of high nutrient content in the fruit, or large sized fruit, or more tastable. So like or more fruits provided by a plant. So like a number of characters we have selected. So those desirable characters which are present in the selected parents. But these selected parents which are always selected the plants present in the wild condition, not nurtured condition. So if the presence of nurtured the wild condition, so those characters which are easily adapted to the environments. So that's why we have selected the parents in the wild nature. So during which we are selecting the two parents, those one act as a male parent and another one as a female parent. So in which, what is the process like, the two events like that is emasculation and bagging process are very important or successful. That is, emasculation is nothing but removal of anthers, from unopened flower buds. Unopened flower buds. Removal of anthers from unopened flower buds. Such phenomenon what we are called as emasculation. So here, while removal of anthers means the stamens or anthers which are removed by a sterile forceps are using. By the using of those forceps, removing of anthers, so just we open, that is corolla part, remove that is corolla, and also we remove the anthers. Why we remove the anthers? So because at the time of while we are using as a desired male parent, so that male parent has a desired character. So such male parent, we are collecting the pollen grains, those are crossing to the female one, because to get the desired characters or desired food product or desired product. So for that, so removing of these anthers, and once again what we are collecting the desirable pollen grains, and those are crossing, those are fused. So that's why removal of anthers from unopened flower bud, it's also very important. What is occur after opening a flower? So if the flower is opened, the mustard should bursting the anthers. That is the maturation of anther. While bursting of anther, they release pollen grains. If they release pollen grain, then mustard should cross them. That is, pollination is carried. So pollination agir. So adhe pollen grain hogi fertilization agir. So illeno self pollination prevent madbek now. So adike emasculation is one of the important process for these. That is artificial hybridization. So after removing of these anthers in the flower bud, that is, the next process is bagging. So bagging is nothing but emasculated flower covered with a paper bag. So that is maybe butter paper bag. So emasculated flower covered with a paper bag, or that is the butter paper bag. It is covered. So for example, it is also, that is paper bag with particular size, with particular size. ಫ್ಲವರ್ ಚಿಕ್ಕದಾಗಿದ್ದು ದೊಡ್ಡ ಕವರ್ ನೀಡಿ ಅಥವಾ ಫ್ಲವರ್ ದೊಡ್ಡದಾಗಿದ್ದು ಚಿಕ್ಕ ಕವರ್ ಹಾಕಿದ್ರು ಕೂಡ ಸರಿ ಅನ್ಸಲ್ಲ. ಸರಿ, that is the thing, but here maybe of the entry of pollen grain. So that's why emasculated flower covered with a butter paper bag with particular size here. So as after it is a paper bag. So these paper bag is tied. So after next we have collected the desirable pollen grains. The desirable pollen grains are collected. Those desirable pollens we have to crossing. At the time of crossing, remove these paper bag and make it crossing or pollination. After crossing, that emasculated flower is once again rebagging. So first of all, why we need bagging? Bagging एक बेकिंग. So just we remove the anthers. There is a stigmatic surface. That stigma is contaminated with the other pollen grains, or that stigma is pollinated with the other pollen grains. Then automatically it is wasted. So that's why to prevent the contamination of other pollen grains, we have to making the least bagging. So after crossing, once again rebagging. So why we need that is rebagging? So sometimes if the stigma is not completely matured, we are making the crossing process, we are leaving, we are open, we are not covering that. So during which any other pollen grain is fall under that is not desirable character. So during which that process is little bit lost. So that's why once again the rebagging process is occurred after the crossing. After the crossing, the bag, that is after emasculated flower is covered with bagging. At the time of crossing, we are open, remove the bag and make it crossing. Once again rebagging is must and should the same what we are using the butter paper bag. So as like these two processes, that is emasculation and bagging, are the successful events or successful process for these successful, that is artificial hybridization process. So these process who are using a plant breeder, who prepare, that is the desirable plants. So that is the plant which consists of desirable characters. So next, so these totally belongs to pre-fertilization structures and events. So in this chapter, we discussed pre-fertilization, fertilization or double fertilization, and post-fertilization structures. So we complete that is pre-fertilization structures and events. So next, that is double fertilization. Double fertilization. So double fertilization, it is first discovered by S.G. Nowosky. S.G. Nowosky in 1898, who discovered double fertilization in flowering plants. What is fertilization? Fertilization, that is nothing but fusion of male gamete with female gamete. Male gamete with female gamete to form zygote. that is fusion of gametes, what we are called as fertilization. So here, what is double fertilization? Double fertilization it is only in angiosperms. There is two types of fusion of gametes because already we discussed that there is a pollen tube which carried the two male gametes and vegetative nucleus. These two male gametes which fused with two different components or two different cells of embryo sac. So that is the first male gamete, that is the first male gamete fused with egg cell. The first male gamete fused with egg cell to form diploid zygote. So this phenomenon is called fertilization, or it is only called as syngamy. So it is called syngamy. So as in the embryo sac, so write only the embryo sac. This is the chalazal pod. It is the micropylori end. As it consists of three antipodals. So here the egg cell. A pollen tube carried two male gametes. One of the male gamete which fused with the egg cell, it leads to formation of zygote. The first male gamete fused with egg cell. It leads to formation of diploid zygote. Such phenomenon what we have called as syngamy. The first fertilization is called as syngamy. As the second male gamete ಇಲ್ಲಿ ನೋಡಬಹುದು ಇದು first male gamete ಇದು it fuses with the egg cell. This is second male gamete. The second male gamete fuses with the central cell or polar nuclei. So in the diagram, the first male gamete fuses with the egg cell. As it is, it leads to formation of a diploid zygote. The second male gamete, this is second male gamete fuses with the central cell. That is what we are called as the second male gamete fused with fused with central cell or polar nuclei. These polar nuclei is diploid. The polar nuclei, it consists of two nuclei. Those nuclei which is formed by opposite poles. So those two, one nuclei from class-7, one nuclei from micropylar, it leads to fusion to form central cell. The central cell consists of two nuclei, hence it is diploid nature. Fused with central cell, it is the diploid. To form triploid pen, that is primary endosperm nucleus. It leads to formation of triploid primary endosperm nucleus. These primary endosperm nuclei is 3N in nature, that is triploid nature. So these two processes, such phenomenon called or such process called triple fusion. Like syngamy plus triple fusion, syngamy and triple fusion is called double fertilization. So double fertilization is nothing but syngamy and triple fusion is called double fertilization. So by the double fertilization, there is the two products are formed. So the two products are nothing but the first one is that is zygote, and the second one is that is endosperm, or that is PEN, that is primary endosperm nucleus. These two are the products of double. So the double fertilization, it is present only in the angiosperms. Angiosperms are the characterization of these double fertilization. Only the flowering plants possess these double fertilization. So once again, the double fertilization, it is discovered by S.G. Navastin in 1898 in angiospermic plants. So what is double fertilization? Syngamy and triple fusion both combined, which are called as that is double fertilization. What is syngamy? The first male gamete fuses with egg cell. The second male gamete which fuses with the central cell or polar nuclei. It leads to formation of primary endosperm nuclei. So such phenomenon what we are called as triple fusion. So these two types of fusion, the two male gametes which fused with the two different cells or different components of that is embryo sac. So that is what we are called as double fertilization. So next, post-fertilization structures and events. Post-fertilization structures and events. Post-fertilization structures and events. So just now we discussed that is double fertilization. In double fertilization, the two products are formed. The two products are nothing but that is formation of zygote. Formation of zygote as formation of primary endosperm cell. Primary endosperm nucleus. So after the ovules developed as, so after fertilization in the embryo sac, the entire ovule which developed as seed. As the embryo sac present in the ovary is developed as fruit. So these all are the events of post-fertilization. After fertilization, the zygote is formed by syngamy. Primary endosperm nucleus it is formed. It is the triploid by triple fusion. After fertilization, syngamy and triple fusion, the ovule is developed as seed and the ovary is developed as fruit. So these are all events which comes under the post-fertilization structures and events. So here the first we discussed as that is embryo. Here, first we discuss that is endosperm. How the endosperm is formed? So means primary endosperm nucleus developed into endosperm. So how the development takes place of development of endosperm takes place by the primary endosperm cell. So as in the embryo sac, previously what we say is that is the central cell. It has a polar nuclei. So these polar nuclei, which is fused with the second male gamete, it leads to form of primary endosperm cell. These primary endosperm cell, it is triploid nature because it consists of 3 nuclei, fusion of 3 nuclei. These entire primary endosperm cell, which is divide to form a mass of cells in the embryo sac. Especially it is present just below the egg cell or just towards the micropylar end. So as based on the development of endosperm or the divisions occurred in these cells, it leads to formation of a mass of cells called endosperm. A mass of cells present in the embryo sac, it is called endosperm. So the endosperm is a nutritive tissue. Because it supplies the nutrition to the development of embryo sac. Development of embryo. Endosperm is a nutritive tissue which nourish the embryo, during its development. During development of embryo, it absorbs the food through a suspensor or through a haustorial structures. Those haustoria which suck that is food nutrients from the endosperm. As the endosperm is called as nutritive tissue, it supplies the food for developing embryo. So there are, at the time of formation of these, that is endosperm, there are three types of endosperms are formed. Three types of endosperm formed. So these three types, based on formation of cross walls after division of the nuclei of these primary endosperm cell. So those three types are, first one, nuclear endosperm. Second one is cellular endosperm. And third one is elobial. Elobial endosperm. What are the three types of endosperm? Nuclear endosperm, cellular endosperm, elobial endosperm. So the third one is need not to our syllabus. So in our syllabus there is presence of the nuclear endosperm and cellular endosperm, but we discussed because of our improve the knowledge. So what is nuclear endosperm? For example, it is the embryo sac, it is the triploid primary endosperm cell. The primary endosperm nucleus, it is the PEN, primary endosperm nuclei, divide, but it is not followed by cross wall. ಇದೊಂದು ಸೆಲ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ, ಇದನ್ನು ನಾವು PEN ಅಂತ ಕರೀತಾ ಇದ್ದೀವಿ. These primary endosperm nuclei which divide one cell ಅಲ್ಲಿ ಎರಡು ಸೆಲ್ ಆಗಿದೆ, ಒಂದು ಸೆಲ್ ಅಲ್ಲಿ ಎರಡು ನ್ಯೂಕ್ಲಿಯೈ ಫಾರ್ಮ್ ಆಯಿತು. By repeated divisions in the same nuclei, there is presence of number of nuclei. The number of nuclei is increased due to division only in the nucleus. Nucleus will matra divide ಆಗ್ತಾ ಇದೆ, but there is no formation of cross walls. Cross wall form ಆಗ್ತಾ ಇಲ್ಲ. So here the endosperm is multinucleated. It is nothing but there is no cross walls. ಯಾವುದೇ cross walls form ಆಗ್ತಾ ಇಲ್ಲ, but nuclei is divide to form thousands of nuclei which is present in the endosperm. So for this, the best example, there is a tender coconut, elderly. So at the time of a tender coconut, just you collect the water contains a multinucleate endosperm is there. Because the tender coconut water, which consists a number of nuclei without cross walls, hence it is the best example for these nuclear endosperm. It is also one way called as free nuclear endosperm. ಅದನ್ನು ನಾವು free nuclear endosperm ಅಂತ ಕೂಡ ಕರೆಯಬಹುದು. Free nuclear endosperm. The free nuclear endosperm, it is nothing but the primary endosperm nuclei divide without the cross walls, presence of number of nuclei in that. So in that tender coconut, after some times it may be remain, or otherwise those free nuclear endosperm, it's converted into cellular endosperm. So what is the difference between nuclear endosperm and cellular endosperm? Nuclear or free nuclear endosperm, the nucleus of primary endosperm cell divide without cross wall. There is no cross walls formed, but the nuclei is divide. Nuclei divide ಆಗ್ತಾನೆ ಇರುತ್ತೆ, so there is no cross walls. But in cellular endosperm, so whether a primary endosperm cell which divide, suddenly there is formation of cross wall after division. So these free nuclear endosperm is also converted as a cellular endosperm. So at the time of a tender coconut, it consists of free nuclear endosperm. So the tender coconut, it is developed, it is matured. There is stagnant of surrounding of those inner mesocarp, ತೆಂಗಿನ ಚಿಪ್ ಒಳಗಡೆ ಟೆಂಡರ್ ಕೋಕೋನಟ್ ಎಳನೀರು ಇರುತ್ತೆ. ಸೊ ಆ ನೀರು ಆದ್ಮೇಲೆ ಕಾಯಿ ಬಳಿತ ಅಂದ್ರೆ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತಾ there is formation of coconut meat. Coconut meat or coconut kernel. Coconut meat or coconut kernel, that is ಕೊಬ್ಬರಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. That coconut meat is the cellular endosperm. Coconut meat is the cellular endosperm. So another one type of endosperm is helobial endosperm. Helobial endosperm. So these helobial endosperm, for example, it is the entire embryo sac. The first division is transversely. It is divided two cells. Divide two cells. So it leads to formation of a small micropylar and a large chalarzal. The primary endosperm cell, it is divided first unequally by formation of cross wall to form two cavities, large chalarzal and small micropylar. Large chalarzal, small micropylar. So as sometimes it is maybe it is remaining. So these divide without cross wall formation. ಇಲ್ಲಿ cross walls ಇಲ್ಲದೆ ಮತ್ತೆ ಡಿವೈಡ್ ಆಗುತ್ತೆ. ಇದು ಹಾಗೆ ರಿಮೈನ್ ಆಗುತ್ತೆ, ಇಲ್ಲ ಕೆಲವೊಂದ್ಸಲಿ ಮಾತ್ರ ಡಿವೈಡ್ ಆಗಿ ಉಳ್ಕೊಂಡ್ಬಿಡ್ಬೋದು. 1-2-3 times divide ಆಗಿ ಇದ್ಬಿಡ್ಬೋದು. But here there is presence of free nuclear, but it is cellular. So it is intermediate type. Helobial endosperm is intermediate type. Intermediate type of endosperm. So these intermediate endosperm is the helobial. So as we discussed the nuclear endosperm and cellular endosperm. So here nuclear endosperm, it is also called as free nuclear endosperm because primary endosperm cell nuclei divide without cross wall formation. So without cross wall formation, so those primary endosperm cell divide number of times, it leads to formation of multinucleate endosperm. Multinucleate. only cell, number of nuclei. But cellular endosperm in which there is these nuclear or pre-nuclear endosperm may be converted into cellular. ಇದೇ ಬೇಕಾದರೂ ಕನ್ವರ್ಟ್ ಆಗ್ಬೋದು ಅಥವಾ primary endosperm cell divided that is followed by cross walls ಜೊತೆಗೆ cells form ಆಗ್ತಾ ಇರುತ್ತೆ. So that is the type of cellular endosperm. So these which related to the endosperm. Endosperm is a nutritive tissue which supply the which nourish the developing embryo. That endosperm, it is formed by triple fusion. Don't forget it. It is the product of triple fusion. It is the product of triple fusion. So next, embryo. So next is embryo. So development of embryo from zygote is called embryogenesis. Embryogenesis. So already we discussed that. Development of embryo from a zygote. In the embryo get assured nutrition from the endosperm. As just a minute. So the embryogenesis in both the monocots and dicots. Monocots and dicots. The early stages of developmental stages is same. There is no change. But structurally it is different. So based on the structures of embryo of both dicot and monocot is various. So how those the embryo is formed from the zygote. For example, it is the zygote. So the zygote formed in the fertilized embryo sac. So embryo sac ಅಲ್ಲಿ fertilization ಆಗಿ so zygote form ಆಗಿರುತ್ತೆ. So ಅಲ್ಲಿಂದ ಮಾತ್ರ zygote ನಾವು ಕನ್ಸಿಡರ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. So as the zygote first divided two cells. So it is the basal cell, it is the terminal cell. The zygote divided into two cells. These two cells are, one is basal cell, another one is terminal cell. It is what we are called as proembryo. Madam? Madam? So the zygote which developed into two cells, the two cells which has the proembryo, one is basal cell and one is terminal cell. The terminal cell which is present towards the micropylar end, the basal cell which is present towards the chela cell. As you see here, it is the embryo sac, that is fertilized embryo sac. It is the primary endosperm cell. See here, it is the zygote. The zygote divided, it leads to formation of one larger cell, one smaller cell. One larger cell and one smaller cell. So here, the larger basal cell and the smaller terminal cell. These large basal cell, it leads to formation of 6 to 8 celled hastoria in the liver. It is totally here. These are totally, the basal cell is divided and it leads to formation of 6 to 8 celled suspension, and the terminal cell. These terminal cell, it leads to formation of those divide, that is vertically, and once again they divide longitudinally. It leads to formation of that is globular embryo. So it is a portion of entire globular embryo. The zygote divided into, it is proembryo, it is globular embryo, it is heart-shaped embryo, and it is matured embryo. So the terminal cell which produce entire embryo, the embryo consists of a suspensor. These suspensor present in the embryo, it is called a globular, it is nothing but a spherical shaped round, that is globular embryo. This embryo which is converted as a heart-shaped, so here you are not seeing clearly, it is the heart-shaped embryo. The heart-shaped embryo, it is completely matured, it consists, it is a structure of dicot embryo. So before that, what the early stages of embryogeny, both monocot and dicot are same, but structurally the formation of structures in both dicot and monocots is little bit variant. So just we discussed in next class, that is both monocot and dicot embryo with the remaining portion of that is post fertilization structures and events. Thank you.